one-step approach and the two-step approach. In two-step RT-PCR, the first step involves the reverse transcription, or RT, of RNA by a reverse transcriptase. This reaction generates the first strand complementary DNA, or cDNA. cDNA is then transferred to a separate reaction tube for the second step of this process. During the second step, the second strand cDNA is generated and amplified by a DNA polymerase. So, in two-step RT-PCR, reverse transcription and PCR occur in separate reaction tubes. This approach is used when there's a need to analyze multiple targets from the same RNA sample in separate reactions or to store synthesized cDNA for later use. In the other approach, known as one-step RT-PCR, the reverse transcriptase and DNA polymerase are pre-mixed, which allows the RT and PCR steps to be performed in the same reaction tube. Not only is the one-step RT-PCR approach faster, it also requires less pipetting and minimizes possible contamination, thereby improving data reproducibility. It also enables easier processing of large numbers of samples for high-throughput applications.
Detection of PCR products in real time can be accomplished by using fluorescent dyes or probes. Fluorescently labeled probes detect the amount of specific double-stranded DNA sequences, while fluorescent dyes detect only the amount of double-stranded DNA. Here, we are going to look at the principles behind fluorescent dye-based detection. The fluorescent dye-based method utilizes a dye that emits fluorescence when incorporating itself into double-stranded DNA. The fluorescence signal increases at each PCR cycle as more double-stranded DNA molecules are generated. The most commonly used dye for fluorescent dye-based detection is cyber green. The cyber green dye functions as an intercalating agent that emits detectable fluorescence when bound to double-stranded DNA. As the PCR progresses and the quantity of double-stranded DNA increases, more dye binds to the PCR products and hence signal intensity increases. Since the dye binds to all amplified PCR products indiscriminately, artifacts such as those resulting from primer dimers or non-specific binding of the primers may also contribute to the overall fluorescence. A post-PCR melt curve analysis enables the detection of non-specific PCR products, which melt at different temperatures than the specific PCR product. The fluorescence observed is proportional to the amount of DNA present.
When designing a lamp experiment, 4 to 6 primers are required, specific to identified regions of target DNA or RNA sequence. Amplification initiates from strand invasion by one of the inner primers, and a strand-displacing DNA polymerase extends the primer and separates the target DNA duplex. The first product is then displaced by synthesis initiating from an outer primer which anneals to an upstream target region. As it is displaced, the end of the product forms a self-hybridizing loop structure due to inclusion of a reverse complementary sequence in the inner primer sequence. This annealing and displacement cycle repeats on the opposite end of the target sequence, and the resulting product is a short dumbbell structure that forms the seed for exponential lamp amplification. This lamp dumbbell structure contains multiple sites for initiation of synthesis, from the 3' ends of the open loops and annealing sites for both the inner and loop primers. As amplification proceeds from these multiple sites, the products grow and form long concatenamers, each with more sites for initiation. The result is a rapid accumulation of double-stranded DNA and amplification byproducts that can be detected by a variety of methods.